Hello everyone and welcome to our first module entitled Proteins. This is an educational module with the hope and intent to give you a comprehensive but not very detailed information about proteins. We built this module for two main reasons. Number one, we want to bring everybody up to speed with protein chemistry, and number two is to give you some talking points to have with your prospective NT-Bind customer. So the agenda of this module is first we're going to be talking about the protein and the protein definition, the chemical composition of the protein, their classifications, important case studies, and of course we're going to tie everything to NT-Bind sales. Now first of all, what are proteins? Proteins are large complex macromolecules containing functional group of varying sizes, shape, charge, and hydrogen bonding capacity. If you take a look at the right hand, you will see the great diversity of the different proteins that the scientists are going to be working on. Some scientists are going to be working on very complex protein composed of many different subunits. Others are going to be working with small, simple protein molecules. But at the end of the day, all the protein scientists have spent countless hours studying and characterizing their proteins, and therefore their proteins is very important to them, and they have passion toward the protein that they are working on. Now the diversity of the proteins stems from the different amino acids that it can be made up of. So we have 20 different amino acids. If you look at an average protein, it has 300 amino acids. So any combination of those would yield over 10,000 possible different proteins. Now let's take a look at the protein structure. First of all, we can start by the monomers or the amino acids that the protein is made out of, and is the sequence of the amino acid that protein is made up of is called the primary structure, or you can think of it as bead on a necklace. So each of these corresponds to a different amino acid. Now these amino acids fold into themselves, forming the secondary structure, which is composed of mainly of alpha helices and beta sheets. Now then after that you have a 3D structure of a different combination of alpha helices and beta sheets, and this is called a tertiary structure or quaternary structure. So these are very complex structures composed of many different subunits. To give you a simulation of how we go from a primary structure into a quaternary structure, we have a movie here, a simulation of NTL9. So here's our protein. Here's our amino acid sequence. So this is a primary structure, and as you can see, it's already forming a secondary structure right here. So this is one secondary structure, alpha helix, and you'll see beta sheets right here forming. It looks like in the shape of an arrow. Now one thing we should know is that the hydrophobic region is formed in the middle, and this is being tucked into the protein, and the outside of the protein is a hydrophilic region. Now what's happening right now is protein is folding in itself, and what the protein is trying to do is trying to find out the most thermodynamically stable state that it can have. So it's coming together. The protein is breaking the bonds and creating new ones, and is going to try to find the most thermodynamically favored state. And here it is. This is the most thermodynamically favored stable state, and this, now you have a fully functional protein with a hydrophobic region inside the protein tucked in, and the hydrophilic outside of the protein. This is going to become important in the later modules. Now that was a simple protein. Here's an example of a bigger protein, a larger proteins that's composed of many different alpha helices and beta sheets. Now this protein is in solution, and that's why you'll see the different water molecules on solutes that the protein is interacting with in the solution. So protein always interact with the environment around it, and it's always dynamic, it's always changing its shape and conformation. Now the main thing really that we can get out of this first part of the module is that protein is very complex. Protein always interact with the environment around it, and this is why we have the thickness of the protein. And number three, the hydrophobic domain or the hydrophobic region is tucked inside of the protein. Now, this is the second part of the module, which is basically the different type and classifications of the proteins. So the main classification of protein that we are interested in is the first four: communications, enzyme, protective, and transport. And the reason we are interested in them because the average abundance per protein is low. Now that's of course on average, and then every time we see a low abundant proteins, we know that we can create value to the customer by selling to them antibody. So communication proteins, so basically they communicate or they relay some messaging from point A to point B. So they transmit or carry chemical signals to distant organs or neurons, which in this case would be neurotransmitters. And examples are hormones, for example, insulin, and neurotransmitter, and there are several of them out there. Now, to give you an indication or to give you an idea of why would somebody be interested in studying communication protein, we're going to take a look at insulin and diabetes. Now, every protein is made up somewhere in the body. So the insulin is made in the pancreas, and to be exact, is made in the beta cells within the pancreas. Now here we have the bloodstream carrying glucose. Now glucose is going to diffuse from the bloodstream, is going to go into the beta cells, and then the beta cells, what they're going to do is going to produce and secrete insulin. So here's the insulin is being produced by beta cells and is going to diffuse into the bloodstream, and is going to be carried on to the different organism and the different tissue within the body. So here's insulin traveling within the bloodstream. Now you have insulin and glucose. They're traveling in the blood and they reach a muscle tissue, and then we're going to see what happens then. So this is a picture of a cell membrane, and the cell membrane, they have the insulin receptor are sticking out, and what you're going to see over here is glucose and insulin's coming right here. Now insulin receptors will bind to insulin and insulin only. Once the insulin binds to the insulin receptors, the receptor becomes activated. Once you activate enough receptors, you're going to see something interesting happening. And what it is you're going to have the glucose transport channel proteins. So we'll be discussing transport protein in the later section, but these transport proteins are going to open up or become activated, and then what they're going to do is they're going to take in the glucose from the outside matrix into the cells. So this is why insulin is very important in uptake of glucose. Now, of course, if you have diabetes, so then your body or beta cells is not producing enough insulin, or it's not producing insulin at all. And then what happens is your cell cannot uptake that sugar that is in the bloodstream, and that's why you have high blood sugar. Now, when we looked over hormones and low binds, we found that there are at least 519 different articles that mention both hormones and low bind. So here are some questions that you may ask the scientists that's working on communication proteins. Where the proteins are being made, and that will give you an indication of how hard is it to prepare the samples or look at the protein. And then number two, is a communication protein secreted in the blood or is it organ specific? And we want to find out if it's secreted in the blood, that means it's going to be in higher, most likely it's going to be in higher quantity than if it's organ specific like a neurotransmitter. And then we can also ask them what is the function of proteins in terms of the response it elicits. So what kind of response this protein elicits. And is an overexpression or underexpression of this communication protein or biomarker is an indication of a specific disease. For example, diabetes. So if you look at diabetes and we'll look at the insulin level, we find them is very low. So there are different diseases that has different biomarkers, and researchers and scientists are always looking for these biomarkers as an indication of some disease. And after that, the two golden question is what concentration is your protein usually found in the biological sample, and secondly, how hydrophobic and hydrophilic it is. So the lower or the low abundant the protein is or found, and the more hydrophobic the protein is, then the more easier it is for you to sell NT-bind. Now, the second class of proteins are called transport proteins, and these transport proteins, they involve in the movement of ions, small molecules, or even macromolecules such as protein across biological membrane or even across biological systems. So both of these proteins are membrane proteins, and if you have a membrane protein, then it's going to be very challenging to the researcher because it's going to have a lot of hydrophobic domain, and whenever you have a lot of hydrophobic domain, that protein sticks to anything that it can find. So it sticks to other proteins and also sticks to polypropylene. To give you an example of how this works in your body, we're going to take a look at hemoglobin and oxygen transport. So here is our hemoglobin. It is a complex protein made up of 4 different subunits, and it has a heme group. And what the heme group does is that it really attracts and keep trapped the oxygen molecule. So it binds to it. The color would change from blue to red, and then at one point it gets released back and turn back the color of hemoglobin into blue. So of course, this is very important to your body. And here it is, a transport protein. Hemoglobin keep transporting oxygen to the cells and then extracted from the lungs. Now again, there are over 1,100 journal articles that found combining the terms transport protein and low bind. Again, the question that we should ask our customer is it membrane-bound protein or is it membrane transport protein? And if it is, most likely it's going to be hydrophobic. Most likely the protein is going to be very challenging to study, and most likely you'll be able to do an NT-bind sale. Now we can also ask them what kind of chemical or ion or protein does this protein transport. The next classification of proteins is enzymes. Now, enzymes are macromolecular biological catalysts. So they are catalysts. They facilitate a reaction by lowering the activation energy that is required for the reaction to occur. Whenever we talk about enzymes, there are 3 key terms that we should be aware of. Substrate, which is the chemical that the enzyme works on or hydrolyze, and the active site, that's where the reaction takes place within the enzyme, and inhibitors, so that's any molecule that binds to an enzyme, cause it to change its shape and decrease the activity of that particular enzyme. Now to show you an example of how enzymes work, of course, every enzyme is located somewhere in the body. So this one is sucrase, and it's located in the small intestines in the epithelium layer. Now, this is again to show you that where the protein is being made, it might be a challenge to the researcher to find and purify that proteins. So here's a picture of our enzyme, and I want to bring your attention to here. This is called the active site, and this is where the substrate sucrose is going to come in and bind to the enzyme. This is also called the lock and key configuration, which means that the enzyme will only work on one substrate. Now once the substrate reaches the active site, the enzyme changes shape, works on it, does a reaction, in this case is hydrolysis, and release its outcome or product, which is glucose and fructose in this example. And the enzyme keep going again and again and again with different sucrose molecules. Now, different question that we might ask a scientist who's working with an enzyme is, what is the function of the enzyme that you are working on? What does this enzyme do? What does it break, or what does it hydrolyze? And also, A lot of scientists, what they're doing is they're doing protein engineering to increase the catalytic efficiency of that particular enzyme. So they want to make sure that the enzyme works a little bit faster. So they engineer it by adding or removing certain amino acids within the active site. So a lot of protein engineering comes in play in this to increase or decrease that catalytic efficiency or the rate at which the enzyme is working. Also, we can ask if the enzyme is part of any high throughput screening, and of course our two golden questions: how hydrophobic the protein is, and how abundant is the protein that you are working with. The second class of proteins are protective proteins and basically antibodies. Antibodies is a part of your adaptive immune systems, and what they do is they recognize and bind to a particular antigen, bacteria or virus, and aids in its destruction. Now, antibodies has been studied extensively, and every antibody will have a heavy chain, a light chain, and a variable region. This variable chain or region is the one that recognizes a specific antigen. So for each bacteria, there is an antibody that can recognize it. Now, to see how this works in the biological system, if we see this is a blood flow and we have a virus that is flowing in the bloodstream. Now this virus over here is going to come and attack the cells. So the first thing it's going to do is going to attach itself to the cell, and then going to recruit other enzymes. Other viruses to come in and attach to the cell. Now here we have the antibodies. So again, antibodies. This is the heavy chain, and this is the two light chain. And as you can see, this antibody right here only recognizes this particular part of the virus or the antigen. So it only recognizes this part and only binds to this part. And once it's bound to the virus, two things happen. Number one, the virus is not able to attach to the cell, and number two, it signals the macrophages to come in and clear that virus out. And really, this is the function of the antibodies, the antibodies in the blood. When we looked over the combination of antibodies and loci, we found over 1,000 different journal articles. Some of the questions that we can ask: is it monoclonal or polyclonal? Does it recognize one part of the antigen or many different parts? And also, do you purify or express your antibody in-house, or do you buy it from somewhere? And at what concentration are you using this antibody? And you can also ask them, what is this antibody raised against? What does it recognize? The other set of proteins, they're not going to be too relevant, and the reason is either because they exist in high abundance, or because they're very rare that not a lot of researchers or a lot of scientists are working on. So some of these are toxin proteins, which are protein that cause toxicity to the cell. The second type of proteins is structural proteins, and these proteins, they maintain the integrity of the cell structure, and mostly are keratin and collagen. And again, these proteins, they exist in high abundance, and therefore anti-bind is not going to offer a lot of value to the researcher or the scientists who are working with these type of proteins. Storage proteins, again, they exist in high abundance, and what they do is they act as a biological reservoir of metal ions and amino acids in an organism. One fun fact, storage proteins found in plant seeds account for 25% of the seed's weight. So they store minerals and amino acids. Contractile proteins, this is acting on myosin in your muscles, and then basically the movement of them or their activation causes movement. Again, these proteins exist in high concentration or high abundance, and therefore we're not going to talk about them in details. So lastly, to sum everything up and put everything in perspective, here we segmented our customers based on the protein that they are studying or the protein they are characterizing. Now we're very interested in these four types of protein: communication, enzymes, protective, and transport. And an example for a communication enzyme, communication proteins is hormones, neurotransmitters, or biomarkers. And the type of research is that scientists do on these communication proteins is they try to identify them, quantify them, and characterize them. So finding out what's different amino acid composition of the protein and how do they work. Now they're relevant to us because they are present in low concentration and have a very short half-life. So every protein that exists in low concentration would be a good target. Enzymes, there are a lot of examples, but basically everything that ends with ASE, that means it's an enzyme. So carboxylase, protease, acetylcholine esterase. These are all type of enzymes. And the type of research that's being done on enzymes is to identify the enzyme. You can also modify it to make it work a little bit faster, which is or increase its catalytic efficiency, and also characterize it by finding out what is primary structure, what is the secondary structure, and how does it work in the body. And again, it's relevant because it can function at lower concentration, so it's a valuable protein, and the researcher might use a small amount of it to see the reactions that they're looking for. Protective proteins, examples antibodies, and the type of research is modifications. So they raise antibody against a specific antigen. So for the Zika virus, they already raised some antibodies against it, and these antibodies are sold in a kit for diagnosis. And they're relevant really because they are used either for drug or diagnostic kits, so they're going to be very expensive, and the researcher or the scientist want to keep to himself the maximum amount of protein antibodies that they can have. Transport proteins, example is membrane transport proteins, and again we're looking for identification and characterization, and they're very relevant because they're very challenging proteins to work with. They're very hydrophobic and they stick to everything that they can find. So in conclusion, I hope this module gave you an overview of proteins, the different classes, and the complexity that exists within the complexity that exists in the protein structure and interactions. And understanding this common terminology about protein and understanding the classification of proteins will help you better understand the customer needs and what the customer is trying to achieve. So I hope this module was helpful, and I look forward to our next modules. Thank you.